Welcome, comrades perennials, to another episode of the audio podcast Synthetic. Artificial intelligence will not replace you, but someone using artificial intelligence definitely will. Right now, your skin is acting as this highly acidic, hostile minefield. We often think of our skin as just a passive covering, like a simple physical envelope just keeping organs inside.
Yeah.
But biologically speaking, it is an incredibly active, aggressively hostile immune barrier that's designed to constantly fight off microscopic invaders.
Okay.
It maintains an acidic pH that acts as a chemical deterrent. It secretes a constant wash of antimicrobial peptides, and it deliberately and continuously sheds its outermost layers, specifically to physically remove any microscopic hitchhikers trying to gain a foothold.
I see.
On top of all that, it is heavily patrolled by specialized immune cells acting as a dedicated border security force.
Oh, wow.
Which brings us to a terrifying biological contradiction involving an emerging multi-drug-resistant fungal pathogen known as Candida auris. For anyone tracking global public health, this specific organism represents a genuine escalating crisis.
Yeah.
It was first identified in the external ear canal of a patient in Japan roughly 15 years ago.
Yeah.
And since then, it has triggered deadly, intractable outbreaks in hospitals and long-term care facilities worldwide. The mortality rates associated with invasive infections of this fungus are staggering, and our standard antifungal arsenals are frequently useless against it.
Okay.
So the goal for you, the listener, as we navigate through this scientific investigation, is to understand exactly how this deadly fungus manages to hide in plain sight. You have to consider why Candida auris treats your highly acidic, hostile skin as a permanent, comfortable safe haven, especially when you look at its close biological relative.
Yeah.
Candida albicans is a familiar mucosal organism. It actually thrives in the human gut and the mouth. But when Candida albicans attempts to colonize the outer skin, it is rapidly identified, attacked, and completely cleared away by the localized immune system.
I see.
The fundamental mystery here is how C. auris manages to effortlessly accomplish what its close cousin absolutely cannot, allowing it to initiate catastrophic global outbreaks entirely undetected.
The comparative study utilizing marine models, or mouse models, provides the foundational data to explain this exact discrepancy.
Okay.
Researchers conducted long-term, side-by-side experiments introducing both C. albicans and C. auris to the skin of mice to rigorously track the physical and biological outcomes over time. The divergent results illustrate the unique threat profile of this pathogen. When C. albicans is applied to the mammalian skin, it struggles to maintain any significant or lasting foothold.
Oh, wow.
The host immune system immediately recognizes the organism as a foreign invader. It mounts an aggressive, sterilizing inflammatory response, and the fungal burden drops precipitously within days. The barrier functions exactly as it is supposed to.
Okay, I see.
But C. auris behaves entirely differently under the exact same conditions. It colonizes the skin asymptomatically.
Wait, asymptomatically?
Yeah, this term is critical to understand in a clinical context. Asymptomatic colonization means the fungus provokes absolutely no visible redness, no swelling, and no clinical signs of inflammation that would alert the host or a doctor to an active, massive invasion. Furthermore, it achieves exceptionally high titers, establishing massive, dense fungal populations that demonstrate incredible durability, persisting on the epidermis for months.
Oh, wow.
This silent, high-burden colonization is the critical factor that fosters rapid person-to-person transmission in clinical environments. When a pathogen can live indefinitely on your skin without triggering a single immune alarm, you become an unwitting, highly efficient vector.
Yeah.
You shed the fungus onto bed sheets, medical equipment, blood pressure cuffs in the hands of healthcare workers. More alarmingly, this durable colonization drastically increases the risk of invasive, deadly disease for the colonized individual. The fungus essentially waits in this silent reservoir. It bides its time until the host's internal immune defenses eventually weaken due to illness, or until a physical breach like the insertion of an intravenous catheter or a surgical incision provides it with direct, unimpeded access to the bloodstream.
Wait, let me make sure I'm fully grasping the mechanics of this contradiction for the listener.
No.
Our skin is quite literally designed to be a barrier that aggressively evicts squatters. If the immune system is actively spotting one version of this fungus and deploying a massive biological strike to wipe it out, the system itself isn't blind or broken. The patrols are clearly out there looking for threats.
Yeah.
So if C. auris is sitting there by the millions without triggering any alarms, you have to look at exactly where it hides on the skin and what specific armor it deploys the moment it arrives.
Okay.
Think about an infiltrator crossing a border into a distinct, highly guarded territory. They do not wear the same uniform they wore in their home country. They step across the line, immediately analyze the local weather and the local customs, and they swap their recognizable garments for highly specific camouflage, tailored to that exact environment.
I see.
To understand how the fungus pulls this off, you have to look at your own arm right now. The skin is not a flat, uniform sheet of tissue like a piece of paper.
Yeah.
It is an incredibly varied, complex topography characterized by sweeping valleys, microscopic ridges, and deep structural invaginations.
Okay.
For a microscopic organism, moving from a cold, dry hospital bed rail onto a living human arm is a massive, traumatic shift in climate. The fungus encounters sudden, extreme changes in temperature, salinity, and available nutrients.
Mm-hmm.
The infiltrator has to find a secure bunker somewhere within this three-dimensional landscape, and then rapidly deploy a specialized defense mechanism triggered by the specific physical conditions of that new hostile environment.
The specific topography of this colonization was revealed through the application of advanced volumetric quantitative confocal microscopy.
Wait, volumetric what?
Volumetric quantitative confocal microscopy. To translate that methodology, this imaging technique allows scientists to visualize the skin not just as a traditional flat microscopic slide, but as a fully realized three-dimensional landscape.
Okay.
They map the depth and physical structures of the tissue. This 3D imaging demonstrated that C. auris exhibits a profound tropism to hair follicles. It does not distribute itself randomly across the flat, exposed planes of the epidermis. Rather, it possesses a direct, specialized hair-binding activity, actively migrating and anchoring itself deep within the physical invaginations of the hair follicles.
I see.
It finds the deepest bunker available in the landscape. To understand the environmental trigger responsible for its survival in that bunker, researchers had to replicate the precise physical environment of human skin in the laboratory.
Okay.
They developed a highly specialized culture medium utilizing synthetic sweat. This synthetic environment is characterized by intense, high salt concentrations and a notoriously nutrient-poor profile. And they maintained it precisely at 37 degrees Celsius to mimic human body temperature. This laboratory simulation proved that the physical environment of human skin acts as a direct mechanical trigger for the fungus. Under these exact physiological conditions, and only under these conditions, C. auris actively remodels the fundamental architecture of its cell wall.
Okay.
It is not a passive survivor simply enduring a harsh climate. It is a highly reactive organism that physically senses the distinct chemical and thermal signature of mammalian sweat and structurally reorganizes its outermost biological boundaries in a calculated response to that specific microclimate. If the fungus is actively remodeling its outer wall the moment it senses human sweat and body heat, you have to question the nature of that new camouflage.
Yeah.
Is this physical remodeling simply making the fungus invisible to the immune system, or is it actively projecting a specific biological decoy to manipulate the body's natural alarm?
If an invader hides in a bunker, throwing a simple invisibility cloak over the door is one strategy. The Border Patrol walks past, they see nothing, and they keep moving.
Okay.
But a far more sophisticated, dangerous strategy involves placing a counterfeit beacon outside that bunker, a beacon that deliberately broadcasts a highly misleading biochemical signal, forcing the patrol to deploy their defensive resources to the wrong sector.
Yeah.
Or worse, tricking them into turning off the perimeter defenses entirely.
I see.
We have to consider the specific molecular nature of this cell wall change. It dictates whether the fungus is merely trying to blend quietly into the dark shadows of the hair follicle, or if it is engaging in an active biochemical sabotage campaign directed against the host's surveillance network.
An analysis of the divergent molecular strategies between the two fungal species reveals that this is absolutely a campaign of active biochemical sabotage.
Oh, yeah.
Fungal cell walls are not simple, static shells. They are highly complex, dynamic structures composed of various overlapping layers of polysaccharides. And the specific arrangement of these complex sugars on the outermost layer dictates exactly how the mammalian immune system responds to their presence. Under skin-like conditions, the easily defeated C. albicans exposes a specific structural sugar called beta-1,3-glucan on its outermost cell surface.
I see.
Beta-glucan is a highly conserved molecular pattern across the fungal kingdom. Over millions of years of evolution, mammalian immune systems have evolved to recognize exposed beta-glucan instantly as a definitive, undeniable signal of a fungal invasion.
Yeah.
When the skin's immune cells detect beta-glucan, it triggers an immediate, aggressive, and highly lethal counterattack.
Okay.
C. auris executes the exact opposite strategy. In direct response to the synthetic sweat and the 37-degree body temperature, it actively suppresses the display of beta-glucan.
Wait, it suppresses it.
Yeah, it hides the universal alarm trigger deep within its cell wall.
Yep.
Simultaneously, it drastically increases the display of an entirely different sugar called chitin on its outermost layer.
Okay, chitin.
Chitin is a tough, nitrogen-containing polysaccharide. Biologically, it is most commonly associated with the rigid exoskeletons of insects and crustaceans.
Oh, wow.
This extensive, calculated cell wall remodeling emphasizes that this is not a simple strategy of invisibility. By deliberately hiding beta-glucan and aggressively exposing chitin, C. auris is projecting a very specific, highly unusual structural sugar into the hair follicle.
I see.
It is a deliberate biochemical decoy that forces the host's localized immune system down a completely different and ultimately flawed immunological pathway.
Think about how a modern building's emergency fire system operates to understand the biological error occurring in the host's body here.
Okay.
In a normal scenario, you pull the fire alarm lever, the sirens immediately ring out to warn the occupants, and the overhead sprinklers deploy water to isolate and drown the threat in that specific room.
Mm-hmm.
What this fungus is doing is essentially hacking that emergency infrastructure.
Yeah.
It pulls an alarm that has been deliberately rewired to shut off the building's sprinklers, allowing the fire to safely burn and spread inside the walls.
Yeah.
The host's immune system is not ignoring the fungus in the hair follicle. It is actively responding to the chitin alarm. The biological sirens are ringing, and the localized immune system fully believes it is executing a rigorous defensive protocol.
I see.
But the specific signal generated by that chitin alarm has been weaponized by the pathogen. Instead of releasing the biochemical water needed to extinguish the fungal threat, the system initiates a cascade that fundamentally shuts down the precise mechanisms required to protect the skin barrier. The invader is using the host's own rigid, hardwired operating procedures against it, turning a highly evolved, localized defense network into an instrument of its own long-term preservation.
A detailed, step-by-step breakdown of these divergent immune pathways illustrates the sheer precision of this biological hijacking.
Okay.
When the typical fungus, C. albicans, is detected via its exposed beta-glucan, it triggers what immunologists call a host-protective type 317 immune response.
Type 3-7.
Yeah, this specific defensive pathway is driven heavily by interleukin-17A, a powerful cytokine. Interleukin-17A acts as an aggressive signaling molecule that commands the surrounding skin tissue to rapidly produce antimicrobial peptides.
I see.
Which results in the fast, sterilizing clearance of the fungal cells we discussed earlier. By contrast, the chitin deliberately exposed by C. auris triggers a completely divergent defensive cascade, a localized type 1 immune response.
Wait, type 1?
Yes. Within the highly specialized, confined microenvironment of the hair follicle, the immune system orchestrates a rapid expansion of very specific cellular players. First, type 1 conventional dendritic cells gather the chitin antigen. You can think of dendritic cells as the intelligence gatherers of the immune system. They collect the foreign material and present it to the T cells. This presentation leads to the massive proliferation of type 1 cytotoxic T cells and T helper 1 immune cells within the follicle. This specific consortium of immune cells leads to the overwhelming localized production of a completely different cytokine known as interferon-gamma.
Okay.
This is where the crucial biological paradox occurs. In many other physiological contexts, such as a deep systemic blood infection, interferon-gamma is a highly protective hero molecule.
I see.
It aggressively marshals macrophages to hunt down and destroy circulating pathogens. However, in the specific microclimate of the hair follicle, the context changes entirely, and interferon-gamma becomes the host's undoing.
Wait, how?
The interferon-gamma produced in the follicle signals directly to the keratinocytes. Keratinocytes are the primary structural building blocks of the skin's epidermis. They are the physical bricks that make up your barrier. When these keratinocytes receive the heavy interferon-gamma signal, they aggressively downregulate the expression of genes and downstream programs that are normally driven by interleukin-17A. By forcing the production of interferon-gamma, the fungus effectively forces the keratinocytes to turn off their own antimicrobial defenses. It actively weakens the skin's physical barrier function from the inside out.
Ah,
The host immune system's production of interferon-gamma directly licenses the long-term survival of the fungus, perfectly engineering a protected, immunosuppressed niche where the pathogen can thrive indefinitely without ever facing clearance.
hmm.
If this incredibly deadly organism relies entirely on this one specific chitin lever to manipulate our localized immunity, could disrupting that single cell wall component be the key to neutralizing its threat entirely? We are looking at a highly specialized, almost precarious mechanism of survival. The entire catastrophic cascade, the type 1 immune response, the cellular expansion of those specific T cells, the localized flood of interferon gamma, and the subsequent fatal shutdown of the keratinocyte defenses is entirely dependent on that initial physical exposure of chitin in the hair follicle.
Yeah.
You have to consider the massive therapeutic potential of targeting that specific molecular vulnerability.
If we can find a way to chemically or genetically prevent the fungus from successfully displaying chitin when it encounters the salty, warm microclimate of human skin, we completely strip away its ability to pull that rewired fire alarm.
Nice.
We force it to expose its true, vulnerable nature to the immune system, allowing the body's natural sprinkler system, that interleukin-17A pathway, to function exactly as millions of years of evolution intended.
The mechanics of this exact vulnerability were definitively confirmed through extensive genetic and pharmacologic manipulations of the fungal chitin detailed in the research.
Okay.
In the study, researchers utilized heavily targeted drugs and highly precise genetic deletions to fundamentally alter the synthesis and physical display of chitin on the outer cell wall of C. aureus in living models. When the scientists successfully altered or reduced the chitin display, it directly and immediately changed the skin's immunological polarization. Without that overwhelming chitin signal present in the hair follicle, the host immune system did not default to the destructive, counterproductive type 1 interferon gamma-driven response.
I see.
Instead, the immune system reverted to its natural protective state. It recognized the pathogen properly, mounted the correct defense, and the sustained asymptomatic fungal persistence within the hair follicle was entirely prevented. This connects directly to the foundational biology of the human meriotissues. The hair follicle on your arm is not merely a biological pore designated for hair growth. It is a highly critical, incredibly sensitive site of constant microbial-immune crosstalk.
Okay.
The dense, complex network of immune cells surrounding every single follicle is constantly sampling the environment, constantly checking the chemical signatures of the microbes living there. This research definitively proves that pathogenic microbes can deliberately chemically bias our epithelial immunity. They possess the immense evolutionary sophistication required to use a fundamentally simple structural sugar to completely reprogram the host's localized defenses, actively engineering a permanent, invisible home on our bodies by turning our own cellular communication networks against us.
If a single molecular shift, just exposing a layer of chitin, can so thoroughly reprogram our localized skin defenses and completely shut down our innate antimicrobial barriers, you have to wonder what other silent, invisible negotiations are constantly taking place in your hair follicles right now, between your own immune system and the billions of other microbes that make up your skin microbiome.
Subscribe to the podcast series Nuclear Locomotive, Neural Sawdust, and Synthetic. Namaste, thank you, and good luck.